Wonderful Fishing Invitational. I just wanted to start out by saying that this is a brainchild of someone who you'll hear from later, Sam Barnard, and he and Mike Sullivan have done a wonderful job hosting the event. And most of all, I want to say, starting out, thank you to Sal from here, from this restaurant, who has hosted us for this event. And I know you all know him well, and you will patronize his establishment as well as Partners Village Store, which provided the lunches today. I'm going to tell you a little bit of a story about the Westport River Watershed Alliance that kind of does me good. Almost 50 years ago, a group of citizens heard about a sewage lagoon being proposed to put into the east branch of the Westport River, and they got together and said, hell no. And the Westport River Defense Fund got together and actually blocked that from occurring. So otherwise we would have a very different river than what we have today. So if you can imagine that. Soon after that, they decided that they needed to actually become an organization, and that was when the Westport River Watershed Alliance was born. For those of you that don't know much about us, we've continued to do advocacy since then, working with the town, and we've continued to do education. We're embedded in local schools, working with children from pre-K right up through high school. The high schoolers just finished up our pollinator garden a couple of weeks ago, helping us with that. And the last thing is that we do water testing and science projects, and you're going to hear a lot more about that. But we have one of the longest running water quality testing programs in the state of Massachusetts. And a lot of you here, just raise your hands, who do the water testing. Pam, I know you do. Or did. Any of you who've been out there in the mornings checking out the DO and all that, we really appreciate you, because we couldn't do 15 sites every Wednesday of all summer without your help. So we thank you for that. I'm not going to talk too much more, but I'm here to answer questions later. And next year is actually when we celebrate the 50th anniversary of the Watershed Alliance, which is kind of a milestone for the organization, and we've come a long way since we began to say hell no about the sewage lagoon. So we thank you for your help in making this all happen. I'm going to now ask Mike Sullivan, our board president, to speak for a couple minutes. We're going to try and keep this all short and sweet, and then you're going to get your dinner, and we won't talk to you during dinner. You have a little peace and quiet, and then we have a presentation by Scott Bennett after dinner. Okay? So that's just what our profile is for this evening. Thank you so much.
Thank you, Deborah. Sam, where is Sam? Where is he hiding right now? Sam, come on up here. I'd like to thank Deborah, and I'd also like to thank Jeff Fitton. Where's Jeff? Jeff, raise your hand. Jeff Fitton is our newest employee at the Watershed. He's our finance and development person, and he's done a great job this year helping Sam, really helping Sam organize this thing. So thank you, Jeff. Appreciate it. Thank you, Deborah. I also wanted to thank a couple other folks. The oysters that you had tonight were donated. They were donated by two organizations. They were donated by Kieran and Kristen Fennerly from Westport Sea Farms, right across the way here. We're lucky to have two nice restaurants here, right in Westport Harbor. And also we have Toph Jasoon. Where's Toph? And his wife Rachel. Now, Toph also has an aquaculture project over here at the mouth of the West Branch. So he works with the Finnerleys. They sell his oysters, and you had some of his oysters here as well tonight. So thank you so much, Toph. Appreciate it. Now, the oysters didn't just jump out of the shell, right? Where are our shuckers over there?
Oh, yeah.
I see Ron Savaria, he's from our Harbor Advisory Committee, or our Shellfish Advisory Committee, I'm sorry. And we got Rick Smith over there. Now, Rick is one of our assistant harbor masters, right, Rick? And we also, and he's on the advisory committee, Harbor Advisory Committee. And then where's Gary Tripp? Where is he? Oh, okay. And Gary Tripp is also one of our assistant harbor masters. He also helped shuck tonight. So how about a hand for these guys and all the work they did? Oh, proud of them. Thank you so much, guys. So I'm just going to piggyback a little bit on what Deborah said about the three tenets of our operation, which are education. No, you stay right here.
Good try.
Well, this is very short. You know, we talked about education, advocacy, and science. And I wanted to just tell you two new initiatives that you all helped finance last year. And this is our educational river tour, something that we wanted to do for years. And last year you got to meet Bill Chase. He's going to make a cameo appearance tonight, and I have a special announcement about that later. But Bill has insurance, he has a boat, and he has a captain's license. And he's been taking out people that we sign up at our organization. And these are not people that are in this room. These are folks who don't have the kind of access to the river that you all have. So it's really a nice, very successful program. And we also, a little bit more esoteric, but we've been involved with the Westport Land Conservation Trust. And we've tried to figure out how we can make a difference at some of the farms around town that are proximate to the river, or proximate to streams and tributaries that feed into the river. So a lot of times farmers don't do a winter crop. It's not profitable. It's really just meant to preserve soil. And so we are now providing financial incentive and paying for seed and the actual, this is a special way this stuff gets distributed. But we now have just under 100 acres that are proximate to the river this last year, the pilot program, that now have a winter cover crop and they're not running off into the river. So we're very excited about that. Now, a big announcement tonight. This group, this fishing group from last year has brought a lot of energy and excitement to our organization, opened up our eyes to a lot of parallel paths that we should be involved with. And we have since established our Fisheries Advisory Committee. Sam doesn't know, but he's going to be on the video.
I do.
But this committee is going to be comprised of folks from our board and folks from the fishing community itself. They are going to be advising us, studying, and looking into projects that we can get involved in that are in our wheelhouse as far as water quality and such, but very specific to fisheries. It's going to start off very specific. We're talking about striped bass, and you're going to hear a lot tonight about striped bass. And 50% of the proceeds from this event will be dedicated each year to these efforts. So that's our big announcement for tonight. Now, Sam is the real driver here. I mean, he's got a lot of thank yous to do, but Sam is, if you get in close to Sam and you start to do a trip with him or something like that, you get sucked into this vortex of activities. And it's Sam AI. And Sue assures me there is nothing artificial about his intelligence, okay? This is Sam AI. AI is activity instigator. And Sam is, he's been a real driving force for this thing. So thank you, Sam.
Mike reminds me of a few things. One is that he won this tournament last year, and I actually paid him $1,000 not to win, which he didn't do. And so,
put it in your mouth. I can't hear you. Here we go.
you can't hear me. Better? Okay. And then Toph, back there. You don't know me, but you know me in a white boat, and I drift across your oyster farm. And I'm desperately trying to get into your oyster farm. And you guys give me the finger and you say, get the hell out of here. And I'm like, can I get a sticker for my boat so I can drift over at high tide? I said, okay, will you promise me?
We'll look into it.
Okay, well,
We'll talk to the boss.
okay, well, I'll talk to the boss privately. These fishing things never can happen without the wonderful support of the guides and our kind of boat drivers. We have 4 professional guides here. Ray Jarvis, Parker Mock, Chris Kellingberg, and maybe Drew Tabor. I'm not quite sure if he's there yet. But I want to thank those 4 because they're donating their time. They could actually be out fishing and making $1. And I appreciate your commitment to this. The rest of us who have committed ourselves to the Westport River Watershed Alliance and these initiatives that we're trying to start, I thank you guys from the bottom of my heart. So I'd like to, I'll mention all of your names, and those of you, there you are. I had that, but okay. So would you guys all stand up, and we'd like to give you a really nice warm hand of applause. Please stand up. Tournaments that you want me to speed it up. We have a number of local sponsors. Sal, thank you again for doing this. And Sal not only does the dinner for us, but he also has given us a gift certificate, which I haven't figured out how I'm going to use yet. I'd like to thank Scott from the Bears Den, who donated a fly rod. Scott, wherever you are, could you raise your hand? Westport Marine has donated a motor which is over here, and that motor will go into the gala raffle, but you all see it here. The Back Eddy Restaurant, obviously Sal. The Saltwater Edge, we've got a $200 gift certificate from Peter Jenkins. Zach's Custom Rods. Zach, where the hell are you?
He's here.
Okay, Zach, thank you. It's a very special thing. Zach also guided today, and he was quite under the weather, but you're rebounded. Westport Bait and Tackle, which we all have been to, has given us a rod and reel, and Partners has given us a small token of whoever catches the smallest fish. Thank you all. I think that's my thing. So thanks. Appreciate it. Thanks for being here.
And this is Chris Killenberg right here. Chris is one of our guides. He and Parker Mock have a business. There are 2 guys in town here they have a business called Westport Fly. You'll see them out on the river. And just, I go out and fish, and a lot of us go out and fish, and we think we know what we're doing. I tell you go out with one of these guys, and you spend a little bit of money, spend a day with them, and you really walk away and you learn a lot. So think about, even if you were an accomplished fisherman, think about some of the businesses you've been introduced to today and utilizing those services. So Chris is one of your guides today, and he is also a big driving force on this committee, this effort that we have to try to do something very specific with the funds that we raise, for a big portion of the funds we raise from this event. So with that, I'm going to introduce you to Chris Killenberg.
Thank you. So I'm going to block these cool pictures of squid and fish because I want to hear what I have to say. Thank you, Mike. So last year, after the tournament, a couple months later, we had kind of a gathering to kind of debrief. What do we learn in the tournament? What do we want to do differently heading forward? And a bunch of us said, well, it'd be really great if some of the proceeds from the tournament could go directly into some program to conserve striped bass in the Westport River, because striped bass are in trouble. And the question was, striped bass are in trouble? What do you mean? And I realized then that some of us who fish regularly and are tuned into this know about this, but a lot of people don't. And some of you, even anglers, don't realize that striped bass are threatened, and the general public doesn't know either, but it's a message we need to get out. So let me just kind of outline the problem for you. For a long time, we catch striped bass and we take some home to eat. We put some back in the water. Inevitably, there's a mortality rate. We kill them one way or another. We either take them home or by accident through injury when we do catch and release. Right now, we're killing them at a faster rate than they're reproducing. And so when that happens, there's an organization that puts together rules, and they include size limits and bag limits and all that sort of stuff. And you try to keep that in balance, but it's out of balance now. And the way to think about it is, we talk about fish in schools. So think of a school. You've got upperclassmen, you've got lowerclassmen, you've got preschoolers. It's a continuum, right? We are now benefiting from the last good class of striped bass, the 2018 class of striped bass, who are the big bass that you hear people say they're catching, etc. The problem is we've got very few young bass following along, and so as everybody ages out, we're just not going to have the numbers that we have right now. And it's nobody's fault, really. We already have rules around catch and release and what you keep, etc. Nature plays a big role in this. The reason that we don't have young ones come along is part they've just not spawned well for the last 6 years. But it doesn't really kind of matter why we're here and we've got a problem. So we said, gee, it would be great if we could do something specific in the river to do our part of a larger problem and protect our fish here in the Westport River. What could that be? And so we came up, and so Mike said, well, why don't you form a kind of ad hoc committee to look at some ideas, which we did. So I don't forget, I'm going to read a list. But anyway, that committee was Sam Bernard, Ray Jarvis, Wyatt Cooper, Ryan Kim, Peter Jenkins, Donald Swift, Roberta Carvalho, Jeff Fitton, Deborah Weaver, Mike Sullivan. We got together a couple times, tossed around some ideas, researched it a little bit, and came up with a couple proposals for the Watershed Alliance for the upcoming year that they might use some of these proceeds for. And so it starts out with some education. We'll do some education, articles in River News, articles in the local paper to outline the problem facing striped bass as a starter, right? Then we'll do some education about better catch and release practices. Then maybe, if you're going to keep a fish, at least keep it legal, right? There are rules, abide by the rules, right? So that'll be a portion of it. And then the other thing was, what can we do about the fish in our river specifically? Is there a habitat thing or whatever? And so we said, well, let's first kind of understand our fish better. Who are they, right? Are they just passing through? Do they come for the summer? Some of them certainly stay, but how do we get an idea of what our fish population is, so then we can think about how we can do some things to protect and sustain them. And so last fall, I was watching a podcast that Peter Jenkins from Saltwater Edge had this guy, what Scott is, talking about this program they did in Woods Hole, and it was so cool. And I thought, wow, we could do something like that to understand our fish here. And essentially the idea is they put tags in the fish that have a radio beacon, and there are receivers for those beacons up and down the coast, so you can see when the fish leaves, in this case Woods Hole, where does it go? Does it come back, and how does it behave in between? And I thought, that just is like the coolest thing, and can we do something like that in the Westport River? Can we understand who our fish are? And in knowing our fish, kind of feel a little bit more inclined to protect them, think of ways that we can educate people about them, habitat restoration, all that sort of stuff. But the first start was understanding our fish. So I'm going to introduce Scott and let him tell you all about this program and the cool pictures behind us. And the idea is that we would institute something like this here in the river, that we would put in the infrastructure to receive the signals of the fish that we tag. We would tag fish, and there's an opportunity here too. And so we have your names and contact information, which is tags for fish don't cost that much, but they're a unique ID, and you can, if you invest in a tag, it's going to kind of be your fish, and you can get the story of that fish. Where does it go, right? Where does it go in the winter? Chesapeake Bay, Hudson River, does it come back? You know, how many years do we track it? It's a really cool way to kind of get involved. So in the coming weeks, once we have this whole thing figured out, we'll reach out to you and say, you know, would you like to sponsor a fish or get together with some buddies to sponsor a fish? The more tags we have, the more fish we have tagged, the better the research, the better the information. So that's kind of where we're headed with some of the proceeds from last year and this year into this tagging program. So I'm going to introduce to you Scott Bennett. Scott is, I want to get his title correct, Manager of Marine Research Services at Marine Biological Laboratory in Woods Hole, and he's also the Program Manager and Co-Investigator of the Striped Bass Initiative Research Program, which is the model for tagging that we would use to kind of help us get going in this. So, Scott, where are you?
All right. Good afternoon, everybody. So now that we've all had drinks, dinner, you can listen to me for a little while, maybe nod off. But first, I do want to just thank the kitchen and all the staff here. You guys have done a great job. Dinner was delicious. All right, so I'm going to tell you a little bit about our research program, what we do, what we focus on, and some of the results that we've found so far. When I started getting emails last night about run of show for today, and I was going to be given half an hour, I furiously sat down on my computer and started to delete half my presentation. So hopefully we don't go too long, but I'll work fast. So let me start. And I want to thank Chris and Mike and Sam and Parker for coming down to Woods Hole a couple times and meeting with me. It was really great to meet you guys, and I'm really happy to be here taking part in this. So this first video right here, this poor little squid here is swimming around. This is Eel Pond. So these are two of our striped bass that come to Woods Hole every summer. And the reason that squid is swimming in the water is because at the Marine Biological Laboratory, we collect live marine specimens for scientists to do biomedical research on. So they are called model organisms, and they are basically biological stand-ins for typically human biomedical research. So cells in urchins do the same thing. Neurons in squid do the same thing as neurons in humans, so we can study these animals to better understand ourselves. So we have these collections of live organisms that come into our facility off of our big collecting boats, some of our other small boats that we go collect. But the squid are used for their giant axon to study neuron development, and a lot of time we have a lot of mortalities that come in on the boat, or animals that get used in research labs, dissected, and then they get discarded. And so this population of striped bass has been established in Eel Pond and Woods Hole for a very long time, pretty much before anyone who's alive now can kind of recollect when it started. And that's why they're there, and that's why this whole thing kind of serendipitously works. We have this population of striped bass that is right next to a marine research facility, so it kind of works. All right, so our program, the Striped Bass Initiative, has, at the moment, we have 2 research sites. So Eel Pond in Woods Hole, and then Squid Knocket Pond in Aquina on the Vineyard. If you've ever been to Woods Hole to catch the ferry, maybe you've wandered by Eel Pond. It's not very big, but it does have a lot to offer. So our program, there are 3 main tenets of our research. So acoustic telemetry tracking, which is what we'd be implementing here in the pond. Archival satellite pop-up tracking using archival satellite tags that are attached to the fish and pop off at a certain date, and they collect data for almost a full year to track where the fish goes in a really fine scale. And then stripe aberration identification. So this is really unique. If you've ever pulled a fish out of the water and you notice the breaks are kind of like sinusoidal aberrations in the stripes, things like that. Once the fish reaches maturity, an adult-sized fish, those stripe aberrations are static on the animal. So you can use them as identification markers for years after you've seen it on the fish one time. So this is Woods Hole. Here's the ferry terminal for anybody who's been on the ferry. But this is Eel Pond right here. The really cool thing about Woods Hole is that it is a tiny little village in Falmouth, but it is home to 6 different independent scientific research institutions. So there's a lot of stuff going on there. So some of the critical points here. So this is Eel Pond Bridge. Right? This is a raisable drawbridge. And this channel right here is the only access and egress point to the pond. So the fish that we track have to come in through there, and they have to go out. And then the MBL's Marine Resources Department, where we hold all of our live organisms that we collect, is positioned there, and that's where the majority of our research takes place. Right? And so this is an aerial view of Eel Pond, and if we zoom in a little bit on our research site, this is our Marine Resources Center. This is our dock. This is our 50-foot trawler that we collect squid with. And so what I'm showing you here are the 2 areas where these striped bass congregate, right? And so it's dedicated to these 2 spots. One really interesting thing is that those 2 groups of fish, they don't regularly intermix with each other. And fish that come back year after year, they go back to their same spot. If you've got a geodock fish, one year, that geodock fish comes back. It will, most often than not, hang out there, and the same for the ones that stick to the back. So even though they're less than 100 yards away, they still kind of keep separate from one another. And so that's what they look like. So if you go to the end of the dock and look down at the water, you can practically pet them on the head. I can feed them. I can hand feed them squid if I need to. So they look like, and then behind the MRC, the Marine Resources Center, we have our other group of fish that hang out in pretty shallow water. So behind the MRC is, at its deepest, probably 3 feet. And then at the end of the dock there is probably 20 feet or so. All right. And this is what they come here to do. So we throw squid, the fish eat it. This is a fun thing to do. You can just sit here all day. You just do it. Some people call it a job. All right. So I'm going to kind of just quickly go through these 3 main focuses that we have in our program to kind of describe what it is that we do and what type of data we get from these studies. So acoustic telemetry tracking, and the focus on it is looking at multi-year residency and attrition of striped bass in an enclosed coastal waterway. So Eel Pond, also I should mention, is fully tidal. It's open to the ocean. It's not brackish in any way. It is a marine environment. And this is what the fish look like underwater. We always get some really nice GoPro footage, especially once we start throwing squid in. The fish get really excited and they kind of start swimming around like that. And they will spend the entire summer in that pond. They arrive in May, and they usually depart sometime between October and November. What's that?
That one was tagged.
Oh yeah, so you'll see some of them do have the, in some of these videos you will see some of the tag. All right. So this is the process for our acoustic telemetry tagging. So the tag in the upper lower left-hand corner there, that's one of our standard acoustic telemetry tags. That is a 5-year tag, so that's a tag that has a battery of 5 years, so it'll track that fish, or that it'll continue to ping for 5 years. So those tags are surgically implanted on the fish and then sutured. We have had, I think, almost 100% success survival rate in these tag implantations. So the process works really well, and acoustic telemetry is one of the research tools that people who do fish tracking use all over the place. So Mass DMF uses this, and they tag thousands and thousands of fish over the course of the years. So this is a really kind of standard methodology. And then any fish that we tag with a acoustic telemetry receiver also gets an external loop tag so that we know which fish it is that we've tagged, and that contact information is labeled on a tag in case someone catches our fish. We hope that they release them, but if they look at the tag, it's just another data point. They can say, I caught your fish here at this date. We've only had 2 people catch some of our tagged fish. If you ever catch a striped bass that has a tag on it, release it, because it's probably got a $400 tag inside of it, maybe.
You only tag fish that are outside the slot then?
No, we go on either side of the slot. Yeah, so we've tagged, I think the smallest one we've done is 25, and then we've tagged some really large 36, 37-inch fish. All right, so here's the entire tagging process. Oops, come on, keep going. That video doesn't want to go. Anyways, whoops. All right. So this is also what makes it kind of easy, because the fish just sit there, so we don't have to go catch them. We can literally just throw a bunch of squid in the water, they all come there, and then we just net one out. So we can do 10 or 12 tags in a regular workday. But you can see, so our facility is right there by the water, so we have a necropsy room where we can go in and we can actually do the surgeries and the tagging. The building there has flowing seawater through it, so we pump water in from Woods Hole Passage. It goes into the building, through pipes in the building, it flows into all of our tanks, and then flows back out into Eel Pond. So the entire time these fish are under anesthesia, they're also getting fresh seawater pumped in their mouth and over their gills to make sure that they stay aerated. So the sutures, and then we mark times for when the animal's pulled out, when the tag is implanted, when they're released, and then we just roll them back down to the dock and plop them in the water, and they will then swim away. Good, a little sleepy. So that's the process. All right. And then, so once the tags are implanted, that's really kind of half the battle. So we also have these acoustic telemetry receivers that we put out around the pond. But these receivers are also the ones that are in arrays all up and down the East Coast. So basically anyone who is tagging fish, any species, for whatever reason, we use these acoustic telemetry receivers, and even if our fish swims by another researcher's receiver, it still picks up the signal. And then when that data is downloaded off those receivers, everyone who uses this equipment all belongs to a cooperative telemetry network, and so all the data is uploaded, and then any tag that you have, which has a unique ID, is tied to you, that information gets sent to you. So every quarter or so, we get a huge data push of our fish, where they've been, things like that. So that's how we can track our fish outside of Eel Pond. And so all we do is we just anchor them to the bottom. The receivers sit in mid-water. We use an underwater buoy to keep the receiver vertical, and then just a top buoy so that we can pull them out of the water at the end of the season. All right, so our array in Eel Pond consists of 6 receivers in the pond, 1 in the channel, and then 1 out here on the Hui dock. And that is so that we can know exactly when the fish arrived locally, when they came into the pond. And then the reason that we have so many receivers in such a small area is because what this is showing is the detection range of these receivers. So they all overlap to such a degree that we can make really fine scale detections of movement, right? So if a fish just swims by one receiver, the receiver just says, I saw your fish, right? And that's pretty much it. It just gives a date, a time, and the number of the fish. But if you have an array like this, where a bunch of different receivers can all see the fish at the same time, then you can do calculations to position that fish in different ways. How long it takes the signal to reach one receiver relative to the other. All right. And so what we found with this method is that we have fish that will come back to Eel Pond year after year after year. So we have specifically identified animals that we can essentially look forward to seeing each May or June whenever they show up. But there is kind of a established attrition rate here. So you can see in this graph right here the number, the percent of animals that return over the course of a few years. So one cohort is tagged, and then over the course of a few years, the cohort that was tagged in that season diminishes and doesn't come back. Whether or not they go to new foraging grounds or they get caught is a little unknown. So that's typically what you can see, and this is just the same, demonstrating the same data there. All right. And we can also do things like look at their arrival in Eel Pond and their departure, and we can compare and contrast those year after year. And so one of the things that we found that's really interesting is that the arrivals of an animal don't necessarily dictate the arrival of that same animal to the pond in the following year, but the departure of an animal in a given season in October or November is relatively consistent over the years. So a single fish basically might not arrive at the same time, but a specific fish very much has like kind of like a drop dead date. And like, no, I got to get out of here. I got somewhere to go, and that's pretty consistent, which is really interesting. All right. And so, like I said, so these fish are resident throughout the summer. So they come in and they stay there. They don't leave. They don't go back out and forage. Maybe 1 or 2 over the course of this, what are we on, 5 years, study 5 years now. But essentially this is, so this is just a demonstration of tracking depth of one animal. They are not floating above the water. You just imagine that the axis is flipped there. So that's actually down. But it shows that that animal was present in the pond from, I think probably the end of May all the way to almost the beginning of November.
What's the water temperature in the pond?
It's always maybe a degree or 2 warmer than ambient. So I think the highest it gets is like 27.
Centigrade.
Yeah.
For the rest of us, what's that in terms?
What's 27? Ask Siri. Hey Siri.
Do it.
I think any of that data is impacted by the fact that they're getting fed.
Yeah. So what we start to see is the fish that are in Eel Pond demonstrate these very clear and predictable circadian rhythm patterns. So at night, they're going to deep areas of the pond, and at day, they are right at the surface, right? and that correlates to when people are there, standing on the dock, making noise, attracting, all right? And so this is a really consistent, predictable pattern that we can see here. But if you zoom out and you look at May and you look at October, what you see is that circadian pattern really, when they arrive, it's really disrupted. It's not consistent. And once they get ready to start to migrate again in the fall, that pattern gets disrupted too. So one of the things that we think is that while these fish are migrating, obviously they don't need a circadian pattern. They're swimming, they're trying to get somewhere, and then once they arrive, they kind of trying to figure their space out, and then once they get into kind of a predictable pattern, they're hanging out in the pond, then they can start to establish this kind of circadian pattern. Right.
How many do you tag in a year?
So we typically do 20 fish every other year, because we try, one of the things we don't want to do is tag a bunch of fish one year and then wait for them to come back, look at their behavior, but then collect a bunch of more fish and run the risk of scaring the ones that we've tagged and making them leave early. So it's just a way of kind of making sure the cohort that we've tagged in 1 year, we get at least 1 year of reliable natural arrivals and departures. So, but we typically try and target 20 fish. We've done 60 so far, not including the archival satellite tags that we put on.
Scott, how many fish are actually in the Eagle Pond ecosystem?
It depends. It depends on the year, but typically the fish that hang out close to the building there, you have anywhere maybe like 10 or 13 fish hang out there, and then I'd say probably double that at the end of the dock. And so this is basically what's showing the data that we can collect from that receiver array. So you'll see the little dot here. So this is one fish that we tagged, and then we compiled all the data from the receivers, and we were able to triangulate it over the course of a few days. So when it's bright like this, it's daytime, and then you'll see the screen dim and go dark, and that's night, and then you'll see the animal moving around around the pond. So in the day they are tight. They are tight right there. Then at night they start to kind of move around and forage, and then they really start to, after this third day, they really start to kind of explore some of these other areas. But during the day they are right there.
What's the max depth in your pond?
About 22 feet, and it's right in the middle right here. All right. So that goes on for a while. All right. More fish videos, in case you're tired of looking at graphs. It's always fun.
They got an easy life, huh?
They really do. And so, to your point about being fed, so they are used to getting fed, but the thing is, these fish don't know that they're being hand fed. They just know that they are in a spot where they can get a consistent meal, right? And so when there's a concept in kind of ecology and species biology where called optimal foraging theory, where basically animals have to make decisions between a sure thing meal, even if it's okay, and risking going somewhere else to try and find new food and running the risk of not finding anything. So essentially what these fish have found is that they're just like, I know we can go to Woods Hole and get a good meal, so let's go there. And that will actually tie into some of the things that we're thinking about for the Westport project as well.
Is there a cap on the size of the fish that come back because it's so shallow and the water's so warm?
No, we've never seen.
So they can come back until they're 30, 40 inches long?
Yeah, we got some big fish in that, especially around the dock.
The far,
Yeah, the deeper one. Okay, So like I said, we belong to a cooperative network of scientists who are all using this telemetry technology, the Atlantic Cooperative Telemetry Network, or ACT. And so this image on the left here is showing all of the established receiver arrays on the east coast of the United States. So it's pretty good coverage, and actually that's a little bit old, so there are more than that. But they're constantly changing because people set out arrays, they pull them out. So it's kind of a constantly changing assortment of detection centers there. And then this is just an example of 4 of our fish that we've tagged over the last couple of years and where they've gone. And so you can see the fish that we tag in this hole go all over the East Coast. Usually the cutoff is the Carolinas for the North Atlantic stock, but we've had fish that we tagged here that go all the way up to the Kennebec, swim all the way up. This is Albany up here, and then go down to the Chesapeake. And you can see, so they're color coded too. So the fish that went to the Kennebec and Albany also went to the Delaware River.
Can I ask you a quick question?
Yeah.
I think the Chesapeake and the Hudson River have been identified as 2 spawning areas, major spawning areas.
Yep.
Are there many others?
So the Delaware River gets lumped into those, so that's like the big 3.
Okay.
But there are others. So you can see here there are some. So I put the biggest rivers here on the map.
Right.
But so this is, I can't remember which one is which, but the Housatonic and the Connecticut River there. Yeah. So those are also areas that have some spawning in them. But there are lots of little idiosyncratic areas where fish will go.
And do your fish that you've tagged who are going up into the other rivers, are they staying there for the winter season?
Yeah, so when they leave in the fall, they usually go down. They usually arrive like in the Hudson. I'd say they usually arrive in December. Most of the time, our detections will kind of cut off in December or January, and then we start to pick them up again in April.
And are they
when come
going back
out.
to their natal streams? I mean, is that what they're doing?
Theoretically, so there is site fidelity to spawning areas that we've seen. It's not 100%, but it is typical.
Okay.
Have any of the fish held over in the pond?
In the eel pond?
Right.
No. So we have they are completely migratory. So we've identified that with the acoustic tags. They all leave at a certain point. Our receivers in the pond no longer pick them up, and then they get tracked all up and down the East Coast. So we know that they are migratory.
Any thoughts on what keeps a fish as a holdover?
What's that?
Any thoughts on why a fish would hold over?
It's a good question. Why a fish would forego spawning, and that being the driving factor in a lot of animals. It's tough to say. If they have a good resource of food there, I don't know why they would. So that's another. Our work in Squibnocket on the island is actually a overwintering population.
But some of them in the fall, they actually go north after leaving Eel Pond.
So that one was a fish that went all the way up north during the summer.
Okay.
Yeah, and then came
So
back.
they usually go south after.
But I will say we did satellite tag a fish, and we're still working on the algorithm that calculates its position. But we did have one animal that we are pretty sure left in the fall, swam up to the Gulf of Maine, and was there for January and February before making a beeline back down to the Hudson and swimming up the Hudson.
Wrong way, Roger, right?
So these are some of the other results that we can get from our data. So we can track here the arrivals and departures. So the black bars here are how long they were in Eel Pond. The blue is how long they were away. So fish 5980 here was a 4-year returner, and so this is that fish from 2018 to 2021, and we were looking at its southern routes for these years. And you can see it's fairly consistent, except for 2018. It left in October, was off Long Island November, January, and February of 2019. It was picked up outside the Chesapeake. And then 2019, it basically did the same route, just into the Delaware, and then it chose the Delaware for the next 3 years. But the issue here is that you see a lot of time between these detections, right? And we'll get to that in a second. And so this is another interesting look at their behavior when they migrate. So this is showing 5 different fish that were all tagged together in a cohort in Woods Hole in the same year. And all 5 of these fish left Woods Hole and arrived at the Tappan Zee Bridge at some point. Right? It's always the Tappan Zee Bridge. It's never going to be the
Now the Mario Cuomo. Now it's the Tappan Zee Bridge.
It's like the Gulf of Mexico.
So this is
Sorry, I'm an old.
so this is showing each of the tracks of each of the fish, right? So the Tappan Zee's here, Woods Hole's here. So we pick them up. They're all kind of taking their own way, but you can see that some fish are getting picked up along a lot of these routes, whereas some of these other ones really kind of don't get detected at all. And what's even more interesting is that when you look at the time it takes for these fish, who all leave at relatively the same amount of time, the disparity on when they reach the Tappan Zee Bridge is really extensive. So you've got fish that are leaving one spot that have spent the entire summer together. They leave together, and then they swim and they're going to the same place. But in some cases, right, this one took 26 days and this one took 94 days to reach the bridge. They're going to the same spot, but they're taking very different routes.
Are they going offshore? Is that why they're not being picked up by those?
And then we can also track the north-south migrations. So this is 5368, and so it was tagged in the fall of 2019. You can see it makes its way to the Hudson, and then in 2020, so it makes its way to the Hudson, and then in the spring and summer, it comes back, does the same thing, goes to the Hudson. Now in 2021, all of a sudden, this fish shows up, even though it went to the Hudson, didn't show up in the Delaware the previous fall. All of a sudden, it comes out of the Delaware River and then comes back to Woods Hole. And then again, it leaves Woods Hole, goes back to the Hudson, and then the next summer, leaves the Hudson, bypasses Woods Hole altogether, goes up to the Kennebec River, and then on its way back, comes all the way back down, skips Woods Hole again, and then goes back down to the Delaware. So, fidelity to spawning sites, maybe. Okay, so that's the acoustic telemetry for our migratory eel pond fish. And then we also started 2-3 years ago a collaborative project with the Wampanoag tribe in Aquinnah to study a population of overwintering fish in Squibnocket Pond. And so this was a really kind of mutually beneficial project because we wanted to study the seasonal rhythms of a population of fish that was not migratory. And the tribe was really interested in studying the movements and seasonal behaviors of a predator of the river herring that they are a population that they're trying to rebolster and reestablish. So we got together and we set out an initial kind of study to look at to assess whether or not you had an overwintering population, right? So basically what happens is that the animals from the ocean, right, from Vineyard Sound, come into Menemsha Pond here, and then they swim through this tiny little river here that goes under a road, and the river at one of the narrower points is about those wide here, and maybe like this deep at certain tidal points, and then comes out into Squibnocket here. And that's the herring, right? And it makes sense because if you're a predatory fish and you want to eat, there's no better option than to basically stick yourself in a bottleneck and just wait for a bunch of herring to swim into your mouth. So we went out and we tagged a number of fish there as well. So the same process, acoustic telemetry tag and then external tag to identify them. And so that's our tagging team. And so we are in year 2 of collecting data about animals that spend the winter in the pond, leave during the summer to go forage around Vineyard Sound, and then come back into the pond to spend another winter there. So we've already had a few animals who have spent 2 years in Squibnocket not migrating. And so the question is, why did you overwinter instead of going to spawn? But it begs the question, what if they are spawning, right? So Squibnocket is brackish, so there is a potential for them to be spawning, but we are going to be doing some seining and other collecting in the near future to try and determine if there are actually eggs and any spawning occurrences going on there. So kind of excited about that. All right. So like I mentioned a little bit, one of the issues that we have with acoustic telemetry tracking is that these tracking dots are not necessarily our fish. They are just a map of the receivers that are out there, the receivers that have said, I've seen your fish, right? Because the fish could swim out here, no receivers, doesn't get picked up, all right? And so you see these huge gaps between some of these detections, right? So what's going on between these, right? And that is where our archival satellite tag comes in. So we can track an animal, we can code our tags to track an animal for 300 plus days, and we can get their full migration route, all right? And so this is what it looks like when the animal is tagged. So there is it is a nylon bolt that gets inserted through the tissue underneath the typically the second dorsal fin. So when that happens, there are a set of bones inside of the fish that support that dorsal fin called pterygophores, and they are kind of like fingers like this, and so the bolt goes through those, and then it kind of locks the tag in like that. We've got fish back at Eel Pond now that still have the harnesses from when we tagged them in 2023, and they are some of the fastest swimmers and most voracious eaters. So the tagging that we're doing, so that the harness that we put on the fish doesn't seem to have an appreciable effect on the animals. And we've actually modified those harnesses recently to be less invasive and easier to apply. And so when the fish swims, the tag it's just barely positively buoyant, and that's so when it detaches, it can float to the surface and start talking to the satellite so that we can hopefully go find it, unless it's in the middle of the ocean somewhere. All right, and so then the tracks that we can get from these animals tell a really interesting story. All right. So this is from one of the fish that we tagged with a satellite tag in 2023, and you can see the date up in the corner. So that's February, April, May. And then it comes back to us. Now, so what you're seeing, so these circles here, those are confidence intervals. That's basically saying how confident the calculation is in that where the animal is. So you saw that that orange blob get really big at some points. So that orange block is the 95% confidence interval. So at some of those really big points, it's basically saying we're 95% confident the fish was in the ocean here somewhere, right? Whereas when it's this small, you can say, okay, our fish is in this kind of like localized area. Because the way that these tags work is not with GPS. The tags are collecting light data, so day duration, they're recording temperature, they're recording depth, and they're recording acceleration. And so when you put all those things together and you run them through a likelihood model against oceanographic conditions, you can basically say, okay, if the tag is recording a day length of this duration at this date, then we know it's got to be at a certain latitude, right? And if the tag was recording a certain temperature at a certain date, we can go back and we can look at oceanographic models and basically say, okay, we know that the tag recorded 5 degrees Celsius. It was only 5 degrees in this area. It couldn't have been in any one of these other areas. And it also records depth. So if the fish goes down to, 1,000 feet one day, you can be pretty confident that it's not right close to the shore, right? So you can eliminate areas and basically build this model track by kind of eliminating areas in the ocean where they can't go. So right now we're working to try to really optimize that that code that calculates all these likelihoods. Because as you can see, some of these tracks, it's basically the computer just trying to do its best. So we're trying to smooth that model so we can get a really good representation of where the track is. Because what we've seen is that a lot of these fish, people typically think of these striped bass as coastal migrators sticking right to the coast. But what we've seen in all the fish that we've put these satellite tags on so far is they make these broad excursions to the continental shelf in really deep water. We had one fish that we tagged last fall where the tag popped off here because it reached a crush depth. The tags are programmed basically when they reach a depth in the ocean where it would crush the tag itself, it automatically releases so that you can get some data back. So we know for sure that fish was at the shelf point, and it wasn't because the tag popped up there. Unfortunately, we think that fish was eaten by a very large tuna because you see it performing its normal rhythms, and then all of a sudden it goes down to depth. But the temperature reading is very, very cold, and then as the tag goes down, the temperature goes up, meaning it was in somebody's warm stomach.
Oh.
And then it came to the surface maybe after making its way through a giant tuna. So the big giant bluefin tuna can easily eat a striped bass. Unfortunately, that striped bass, she was one of the ones that had come back to Woods Hole 5 years in a row that we had tracked. I know. And so this is just an example of another. So you can see how large that likelihood can go. And so the red circle is a 50% likelihood. So it's basically kind of a coin. All right. So we've got another one that you see it's basically the same thing.
Would it be possible to make the tags GPS enabled?
They can. The problem is that they need to make the tag bigger to hold the GPS data. They can't log a year's worth of GPS data. You could make one that could fit on like a whale or a shark, but because of the relative size of the tag to the striped bass, we're kind of limited in a certain way. We've thought about double tagging these animals with the acoustic or the satellite tag and a GPS. We're still trying to kind of figure out how to go about doing that. But the other thing that's nice is that you can incorporate those acoustic telemetry detections into the model as anchor points, right? And so if you have an anchor point at a certain date and the model calculates something that's over here, it basically corrects itself and it says, no, the fish had to be here because we know that this is a set point in time. All right, and so the last of the three kind of main focuses is using stripe patterns to positively identify individual fish. So how many people here have ever caught the same striper twice? Caught them 4 or 5 years in a row.
Yep.
And so we can do this by using underwater images, right? So we just put an underwater GoPro on a long stick, and we throw squid in the water, and we basically take, hours of video of these fish swimming around, and then I painstakingly go through this footage. It's actually very nice, kind of meditative, and take screen grabs of really good side profiles of certain fish. The other thing that we do is that when we tag these fish, when we pull them out of the water and we put these tags on, we also take pictures of the left and right hand side of each of these fish. So we have a known documented image of a single fish, and we know what the left hand side and the right hand side look like. All right? So there's a tag one right there. So then what we can do is go back, look at the reference photo, and then go through this footage in another year and look for these fish and look for these stripe aberration patterns, right? And then what we can do is we can put together these images, right? And so these are kind of just a collection of some of the fish that we have, right? And you can see the arrow pointing to the specific aberration in the stripe pattern over the course of several years. Now, one of the really nice things that makes us really confident in this is that the fish that we are using as our benchmark here have all been acoustically tagged, so we know that we know that they are the same fish, right? Because we can visually identify them by the external tag and by the information in the acoustic tag. And so we can go through and we can look and we can see, yep, we have a straight break here and a straight break here. Same thing in 22 and in 23. This was my baby that got eaten in the continental shelf. Unfortunately, she was back for 5 years. But you'll notice that her tagging event was in 2021, and then she came back 22, 23, 24. I went back and I started looking through old archival images that we have, and I was able to find her the year before she was tagged just by her stripe pattern, right? So this has some really interesting implications because if you can identify a fish by a kind of unique fingerprint stripe pattern, right, you've essentially enabled yourself to, you basically given it a natural tag, right? And so I started working with the people who developed the Got One fish logging app, we gave them all of these year-to-year images to start to train a large visual model AI program to identify stripe patterns so that you could theoretically, if you're using the app and you catch a fish, take a picture of it, and then if somebody else catches the fish, a couple months later, they take a picture of it. Basically the algorithm could match the stripe aberration pattern to it, and you could say, oh, this fish was caught, on the Cape in June, and you just caught it in New Jersey in, September, right? And so basically what you've done then is you've tagged the fish, and you'll get these locations of where the animal is without actually having to tag it. Okay. So, with that, thank you guys for listening. This is the largest audience that's ever heard me give this talk, so thank you. The research is funded by the Edwin Barbie Charitable Trust. They've been fantastic funding our research over the last 5 years and allowing us to kind of expand what we are doing. So I also want to recognize the SBI team, Dr. Steve Zotley, who was the one who started this program back in 2013. He's a retired professor from Williams College, and he basically would just come out and look at these fish and count them every day to see how many there were, and he started collecting these piles and piles of data, and eventually it kind of blossomed into what it is now. Dr. Brian Prendergast, who's at the University of Chicago, who's kind of leading the charge on a lot of our satellite pop-up tag research, and the Wampanoag tribe of Aquinnah, Brett Stearns, Andrew Jacobs, Marcella Andrews, and Andrew Karlinski have been really indispensable in making that program function. And that is my son from a few years ago. He likes to go down and feed the bass. He said right after that picture, "Can I stick my finger in the water?" And I said, "No, because they will definitely bite you," especially in July. Right now they're still a little skittish, but July and August, you know, you can put a toe in there and they'll go after it. So. All right. Well, thank you all very much for listening to this.
We have dessert, but I'm going to see if anybody have any more quick questions, maybe 2 or 3 questions we'll take before we...
I'm going to be here for a bit too, so if you want to, yeah.
How many tags have you applied, and how much do they cost?
The acoustic telemetry tags.
Yeah.
So the acoustic telemetry tags vary in price depending on what you want them to do. So they can be an ID only tag. So the little ping, the 69 kilohertz ping that it sends out can just be an identification number. And those are somewhere around like $250. And then you can also add temperature, and you can add depth to those tags, and then that increases the price if you have both of those to $600 per tag. And then it depends on the battery life too. So you can get 10-year tags, 5-year tags, and then you can also do smaller 1-year tags. And so it's all kind of a variation on what you want the tag to do.
One more question?
What's the sex education? Do male and female fish different, and how do they fertilize the eggs and all that stuff? And
So there is no sexual dimorphism in striped bass.
so they do both.
Sorry, no, you can't tell the difference between a male and a female.
But there is a difference.
Yes, so there are male and female striped bass.
So how, when they go spawn up the Hudson River or something, is the male with the female, or?
Not usually. So the way that typically works is that so the females mature at a later age. So the females mature at about 5 to 6 years. The males mature 2 to 3 years. And usually what happens is that when the southern migration takes place, and let's say they go into the Chesapeake, essentially what will happen is males will go in first, and they'll kind of hang out in the main body, and they'll wait for females to start making their way in. The females kind of, I'm not exactly sure how they know when to start making it. I think it's a temperature trigger, but the females will come in, and then they will go into all of the little tributaries and little small waterways. And they go in there, and they basically just like, they are focused. They're not waiting for the males at all. They'll swim in to these areas, dump eggs, and then all the males basically just rush in and try and fertilize them. And then
Okay, so the eggs are out there, and
they're
then they
broadcast
just throw
spawns,
the
yeah.
sperm on them.
Yeah. And that does happen in waves. So there are a few waves of spawning that happen within a single season. Yeah.
Excellent. Thank you, Scott. That was a very really nice presentation. Small token of our appreciation.
Oh my goodness.
I just want, before I hand it over to Sam for our awards tonight, I just wanted to recognize, was Rick Smith still here? No, he's gone. So Rick is training to be our second educational boat driver. The reason that's important is because our first educational boat tour driver, which is right over here, Bill Chase, put your hand up there, Bill. Bill now has a new job, and now we're announcing as of last night, he is our new harbor master, effective July 1st. Thank you, Bill. Sam, all yours.
So I guess I'm the dessert. Bill, thank you for taking on the responsibility of marine advisory
Marine Services, I think, Director of
Marine Services. So you're going to have to give us all a bit of a free pass occasionally.
Assuming you paid your waterway use sticker fee.
Okay.
We want it.
We have a bunch of prizes to give out, and thank you all for hanging out. Zach, I guess, has left. Zach, who was supposed to give us a rod, but he's got one. There's one. So Bill Richmond, who I have skied with, I've played paddle with, I've played pickleball with, and he always seems to win. And so Bill Richmond wins the largest fish on a spin rod at 29 inches. And he also won the most fish on a spin rod. I thought, Bill, you were a fly guy, but I guess not. So if you would come up here, please. I have one of your awards, but I don't have Zach's award. So you're getting, you're gonna shake my hand?
Yeah.
Okay.
You want a picture?
Yeah. Sure.
Oh. Okay,
Let's try that again. Congratulations, Bill.
All right.
Cheers. Great, thank you. So I'll get the other rod for you.
Okay.
Okay?
I get like two words in my word speech are Chris Killenberg.
Well, Chris does have something coming at the end of this. So we had two people who won the largest fish on the fly. I mean, these were not huge fish today, but Tom Ohaus and Rafi Dursakishan each got I mean, I can't believe they have exactly the same length. There has to be something. So if Tom Ohaus and Rafi could come up here, please. Because they were tied, we have two different rods. Since I'm a R.L. Winston Pro ambassador, that rod is kind of more valuable to me, not necessarily to Scott from the Bears Den. So we have to somehow have a runoff here. And so I thought about that. But the best thing I can come up with is drawing straws. So you have to hang on. I have straws. Hang on, just relax. Well, no, like half a year. Okay, so I had another piece here. Okay, so here.
Get a picture of that. Yeah, really.
Okay. He disappears. Okay, Rafi, you're the older guy, so you draw one of those two. Oh, look at that. Okay, that's easy. So, Tom Ohaus. No, it's okay. So this is the R.L. Winston. It's a brand new Salt Max. It's the best rod they make. Scott, where's your rod? Scott? Well, I don't know where it is. Scott from the Bears Den gave us another beautiful rod.
I have the straw.
I'm not sure. You have the straw.
I'm good with the straw.
You're good with the straw. You'll get the rod.
There you go.
Oh, yeah. Thank you. Maybe a picture?
Yeah, sure.
Gentlemen, congratulations. I think last year Tom didn't get much. You've got a reel. Okay, you got a reel from the Bears Den.
Good, okay.
Okay, so continuing, I'm sorry, I want to get this over with because I knew we were about to go home. The most, this is, Tom, can you come back here again? This is ridiculous. Tom wins the most fish on the fly today, and you get a $200 gift certificate from the Saltwater Edge. Okay, I don't have you long.
How many?
Okay. Is that? I will make sure it's on file, so you have to get me to, cheers.
How many fish?
We have five on the fly. I think we're close. So we have this, we have this, this is a board event, right? Just like every famous country club. Last year, I almost dropped it, I know. Last year, Team Menhaden, which was guide Ryan Kim, anglers Tom Ohouse again, and Mike Sullivan won it last year. So we have a new team award this year, and the team award goes to Chris Killingberg. He is a member of the Soul Brothers. And so, Chris, would you come up here? Because you, where are you?
Oh, here.
We'll get down.
It's a team, right?
Well, yeah, okay, bring your team up. Bring your team. Okay. So, do we have all of your members?
No, is Adam still here?
Adam? No? Okay. So, okay, so if Chris would hold that, you will now be historically forever in the Westport River Watershed. Well, I'm not we.
No, standing in for Adam.
I'm standing in for Adam. So anyway, so that's going to go on the board in the Westport River Watershed Alliance. So when people come in there, they'll say, "Wow, what's that cool thing? You too can do this." So Chris, being the lead captain, wins Sal a $200 gift certificate to the Back Eddy.
Wow, cool.
Sal, are you around? Wasn't around. So cheers. Thank you all so much. What a great event. I hope that Scott kind of got us all juiced up about what we're trying to do here, and anybody here can buy a tag and own their own fish. So think about it. It's kind of a cool thing. And Scott, you can maybe help people out with this. Thank you all. Cheers.